An eel-shaped shark emerges from darkness, bending its long body behind a blunt head lined with needle-like teeth. 6 pairs of gill slits ripple at its throat, their folded margins forming a dark red fringe. If you met Chlamydoselachus, anguineus, without context, the silhouette could resemble an animal misplaced from deep geological time. That impression explains its prehistoric reputation. But appearance alone cannot measure evolutionary age or change. The frilled shark is a living species with ancient relatives, not a fossil revived unchanged. Body combines retained ancestral features, specialized deep water traits, and modifications with complicated evolutionary histories. The mystery is therefore not whether time forgot this shark, but why natural selection preserved this particular form. Each feature must be tested against movement, feeding, reproduction, habitat, and relationships to other sharks. Some resemblances to fossils reflect shared ancestry, while others may persist because they still function effectively. Others only look primitive when compared with familiar fast-swimming sharks shaped for open water pursuit. Our investigation separates lineage history from ecological performance, moving from the frilled throat to the flexible spine. It asks what has truly been observed, what anatomy supports, and what remains speculation in dark water. The answer will not diminish the animal's strangeness. It will locate that strangeness within continuing evolution. The opening body is neither a relic nor a blueprint, but a living compromise under present conditions. Frilled sharks occupy deep continental and island margins in parts of the Atlantic and Pacific Oceans. They are usually encountered through fisheries, and direct observation in their habitat remains exceptionally rare. Their cylindrical bodies taper toward a long tail, unlike the muscular, spindle-shaped profile of many active sharks. The head is broad and somewhat flattened, with the mouth opening at the front rather than underneath. 6 gill pairs distinguish them from most sharks, which usually possess 5 pairs. The first gill slits extend across the throat and nearly meet, producing the conspicuous collar. Fringed tissue increases the visual effect, although the common name describes shape rather than a unique breathing mechanism. Water enters the mouth and passes over gill surfaces where oxygen diffuses into the blood. The folds seen outside are associated with the openings, not decorative structures carried for display. When you watch a living individual ventilate, the supposed fossil ornament becomes working respiratory anatomy. The skeleton is cartilaginous, as in other sharks, and the axial column allows pronounced body flexion. Small fins sit far back, leaving much of the trunk uninterrupted by large stabilizing surfaces. This arrangement supports slow, sinuous movement, but likely limits the sustained acceleration of a streamlined pursuit shark. Live recordings show the body undulating and turning with an eel-like appearance. Those observations establish locomotor capacity, yet they do not reveal normal travel speed across undisturbed habitat. Capture stress, temperature change, and shallow pressure can alter the behavior of brought-up animals. Researchers, therefore, treat brief surface or aquarium footage as evidence of movement, not a complete activity budget. Preserved specimens provide reliable anatomy while catch depths and locations outline broad habitat use. Computed imaging reveals internal geometry without cutting every structure, though scans cannot show natural behavior. Comparisons with related living and fossil sharks help identify likely ancestral and derived features. One question concerns the mouth, where hundreds of recurved cusps form pale rows across both jaws. The teeth are individually small, but collectively create a trap difficult for soft prey to escape. Stomach evidence shows consumption of cephalopods and fishes, including other sharks in some examined individuals. Squid bodies are slippery and muscular, so backward-pointing cusps could resist withdrawal after contact. The front-facing mouth may help seize prey encountered directly ahead rather than beneath the head. Its gape can become wide relative to the narrow skull, expanding the capture zone during a lunge. An approaching target supplies visual, chemical, mechanical, or electrical cues, although their relative importance is unknown. Orientation brings the terminal mouth forward, jaw opening exposes the tooth field, and closure entangles compliant tissue. That cue-action adaptation consequence sequence is anatomically supported, but natural strikes have not been adequately filmed. Claims that the shark routinely coils and launches like a snake remain hypotheses, not established feeding behavior. The long body could assist a short lunge, yet flexibility does not prove a specific ambush technique. Likewise, suggestions that bright teeth lure prey lack convincing direct tests. The teeth may simply be visible because pale dental tissue contrasts strongly with the dark mouth. An empty stomach in many specimens further limits reconstruction of typical meals and feeding frequency. Soft cephalopod tissue digests rapidly, potentially making some prey harder to identify after capture. Fisheries samples can also be biased toward animals attracted to bait or occupying trawlable terrain. The safest conclusion is that the mouth retains slippery prey efficiently and supports a varied carnivorous diet. Another question concerns buoyancy and energy use in deep water. The frilled shark's large liver contains low-density oils that help offset the weight of its body. Reduced negative buoyancy lowers the muscular effort required to remain above the seafloor. A slowly moving shark can conserve energy between irregular feeding opportunities. When prey enters range, flexible turning and a broad gape may substitute for prolonged pursuit. This interpretation fits anatomy and habitat, although direct energy measurements are unavailable. The body plan, consequently, looks unspecialized only when judged against the wrong ecological task. For efficient hovering and short-range capture, different proportions can remain advantageous. Evolution can conserve a feature because stabilizing selection removes variants that perform worse in a persistent niche. Conservation never means genetic stasis, and unchanged outlines can conceal extensive molecular and physiological evolution. The frilled shark has survived environmental shifts by continuing to reproduce, develop, feed, and respond. Its reproductive biology makes that continuity especially demanding. Frilled sharks are aplacental viviparous, with embryos developing inside the mother and receiving nutrition largely from yolk. If you examine the reproductive tract, embryos of different sizes can reveal stages that field observers almost never witness. Development is believed to be prolonged, but widely repeated exact gestation claims remain uncertain. Low temperatures and slow embryonic growth support a long period without establishing one universal duration. The mother carries developing young through changing depth, temperature, oxygen, and food conditions. Embryos consume yolk, grow within protective membranes, and are eventually born as independent swimmers. This sequence reduces exposure of eggs on the seafloor, but places sustained energetic costs on the female. Small litter sizes and slow reproduction may limit population recovery after increased fishing mortality. Actual demographic rates remain poorly quantified because captures are uncommon, and age estimation is difficult. Bycatch records can signal encounters, but changing gear and effort complicate trends through time. An apparent increase may reflect expanded fishing rather than a growing shark population. An apparent absence may reflect limited sampling rather than safety from exploitation. Careful monitoring matters because rare deep-water species can decline before ordinary surveys detect clear patterns. The frilled shark also changes how evolutionary history is communicated. Calling it a living fossil can inspire curiosity, yet the phrase encourages an image of arrested development. Every living lineage has existed through the same elapsed time since its common ancestors with other living lineages. Some lineages retain more visible ancestral traits, but all continue accumulating changes and facing selection. The frilled shark's 6 gills, terminal mouth, and body shape form a mosaic rather than a timestamp. If you replace the imagined museum piece with an active predator, each feature becomes a functional question. Future evidence should begin with long-duration observation at natural depth and temperature. Low-light cameras near slopes could document cruising posture, vertical movement, and responses to prey without capture stress. Records of complete feeding attempts could test coiling, lunging, suction, and tooth engagement independently. Detailed scans across growth stages could show how flexibility, jaw geometry, and gill structures change with size. Genetic comparisons with other hexanshiform sharks could refine when particular traits diverged. Standardized fishery reporting could improve estimates of range, seasonal occurrence, and population exposure. Reproductive samples collected responsibly could constrain developmental rates without turning sparse observations into false precision. Environmental data, paired with encounters, could identify preferred oxygen, temperature, and bottom conditions. Those results might show whether the familiar body plan occupies a narrow refuge or a broader moving habitat. They would also reveal how warming and deoxygenation alter usable depth along continental margins. Returning to the first silhouette, the folds at its throat open and close with every exchange of water. The long trunk bends because its living tissues answer muscles, currents, and obstacles in the present ocean. Its teeth do not demonstrate ancient ferocity. They demonstrate a current solution for retaining elusive prey. Its lineage reaches deep into shark history, but no observer has watched its ancestral forms behave. Fossils preserve shapes and hard tissues, while living anatomy supplies motion that must not be projected backward carelessly. The animal looks prehistoric because several conspicuous traits resemble ancestral conditions and differ from familiar modern shark forms. Those traits persisted within a functioning deep-sea ecology, not outside evolution or beyond environmental change. Chlamydoselachus anguineus is scientifically remarkable, precisely because its ancient-looking mosaic remains alive, specialized, and incompletely observed.